Myself, Dr. G. Prasanna, working as an assistant professor in the Department of Genetics and Planning at School of Agriculture, SR University, Warangal. Today, we'll discuss about the most important topic, that is breeding for pulses. In this pulses, we'll discuss about the red gram, black gram, green gram, and soybean. So, these are the lecture outlines. That is, it includes the centers of origin. We'll discuss about the centers of origin of all the four pulses, and next distribution of the species, next breeding objectives, and next conventional innovative approaches, that are the breeding methods for these pulses. First, we will discuss about the introduction about the pulses. Generally the pulses are low yielders than the cereals. So here are the reasons why the cereals are low yielders why the pulses are low yielders when compared to the cereals. So generally, the pulses are having the indeterminate growth habit. Indeterminate growth habit is nothing but generally the plant, there will be no reproductive stage is observed. Only the vegetative stage we can observe. Because of that, there will be no flowering and fruiting is observed. So this is the because of indeterminate growth habit, the pulses are having the low yield. And next is long continued natural selection, and next is cultivation on the poor soil. Generally, we'll cultivate the pulses on the poor soil, generally on the which are continuously not cultivated. On that soils we'll cultivate the pulses. And next is inadequate use of the fertilizers. Generally, the recommended dose of the fertilizers will not use in case of pulses. And next is inadequate plant protection measures. There will not take the proper plant protection measures, like application of some of the fungicides, like that we will not do. That is also one reason why the pulses are low yielders. And next is rain-fed cultivation. Generally, the pulses are mostly cultivated in the rain-fed areas. So this is also one reason the pulses are low yielders when compared to the cereals. And next is photosensitivity. So, and here also the photosensitivity. Generally the pulses are having the photosensitiveness. So these are the some of the reasons why the pulses are low yielders to the cereals. The first one is indeterminate growth habit, and next is long continued natural selection, and next is cultivation on the poor soils, and next is inadequate use of the fertilizers, and next is inadequate plant protection measures, and rain-fed cultivation, and photosensitiveness. So these are the reasons why the pulses are low yielders compared to the cereals. Next, coming to mostly the pulses are having high protein. Generally, the protein from the pulses are incomplete. Generally, the pulses are having the good sources of the lysine, tryptophan, and threonine, but they are very low in the sulfur-containing amino acids like methionine, cysteine, and cysteine, which are more in case of cereals. So the pulses are more. They are more. The pulses are having the more protein content, and they are having the good sources of the amino acids like lysine, tryptophan, and threonine, but they are having the low sulfur containing amino acids, that are methionine, cysteine, and cysteine, which are more in case of cereals. So, and just now we discussed why the pulses are low yielders when compared to the cereals. So these are the main reasons why the pulses are low yielders to the cereals. And next, coming to first, we'll discuss about the red gram crop. Red gram is also called as arhar or tur. Generally, the red gram is also known as PGNP. The botanical name of the red gram is Cajanus cajan. It is having the chromosome number. It is a diploid species having the chromosome number 2n equal to 22, and it belongs to the family Fabaceae. And next is place of origin. It was originated in Africa or Asia, and and the red gram is also having the wild species, that is a Cajanus kerstingi, and next is relatable crossable genera is Rinkosiga. So this is about the introduction of the red gram crop. Next, generally there are two varieties are present in case of red gram. The first one is arhar, and next is tur. Arhar is the scientific name of arhar is, it is a Cajanus cajan variety bipolar. Generally, this arhar is perennial and late maturing, and having the large bushy plant. It is a large bushy plant and having the purple streaked yellow flower. Here you can observe this is a this is a flower which is a yellow color with purple streaks. that is the Kajan variety bipolar, and next is Kajan variety flavus, that is the Tur. It is a short duration variety and early maturing, and the color of the standard petal is yellow, and the pods are green in color in case of Tur. Generally in Fabaceae family, we are having the four different types of petals. There are different types of petals we are having. The first one is standard petal. In the middle, we will be having the standard petal, and the next two are the wing petals, and next two are the keel petals. So the arrangement of the petal is like butterfly shape. So it is called as a Papilionaceae family. So these are the two varieties which are present in the red gram, that is Arhar and Tur. Here this Arhar is Kajanas Kajan variety bipolar, and Tur is Kajan variety flavus. So here you can see the difference between the Arhar and Tur. This Arhar is a perennial, whereas the Tur is a short duration, whereas Arhar is a late maturing, and whereas the Tur is a early maturing variety. And here you can see the difference of the flower color of the flower. Here in both varieties, the color of the flower is yellow, but in case of Arhar, we can see the purple streaks on the yellow color flower. So these are the varieties of the red gram crop. And next, coming to the breeding objectives, the first breeding objective is evolution of long duration, high yielding variety which are suitable for the rainfed to replace the local land races. Generally, the pulses mostly will cultivate in the rainfed areas. Will grow the long duration, high yielding varieties which are suitable for the rainfed conditions. For that, SA1, which was released during the 1940, and CO6, which is a result of the mutation breeding. And next, second breeding objective is to evolve the short duration varieties around 105 days duration, which are suitable for the irrigated or mixed cropping with groundnut. So generally, for mostly the pulses, generally they are having more duration, but we can see some of the short duration varieties which are suitable for the irrigated or mixed cropping with groundnut. Here you can see ICPL 87 variety, which was released from the ICRISAT, and ICRISAT, that is Hyderabad, and next is Vamban, which is having the 110 days duration. And next is breeding for bold grain type with a desirable seed coat color. Generally, the H53C is a long duration variety with a dull white seed coat color and bold grains. And next is breeding for the vegetable type. Generally, green pods with bold seeds are used as a substitute for the green peas, and the perennial types like Attapadi local are used, and BSR1 is a perennial red gram whose green pods are used as a vegetable. Next is breeding for resistant to the pest. Generally, the Heliothis is a major pest, which is a major pest which was present in the red gram crop, and the terminal cluster types are highly susceptible, and all our varieties are highly susceptible to this pest. And next is breeding for disease resistance. Generally, the major disease in case of red gram is sterility mosaic virus. This sterility mosaic virus, it is because of this sterility mosaic virus, the growth of the plant will be indeterminate. Indeterminate growth habit we can observe, and there will be no flowering, and flowering and fruiting we can observe. And next is root rot and blight. These three are the sterility mosaic, root rot, and blight are the most important diseases in case of red gram. And next, wild species, that is the Cajanus caraboides and Cajanus lineata, are having the resistance to this diseases. And next is breeding for high protein content and quality. Generally, in the pulses, the protein content and the quality is very important. So we have to caretake. We have to caretake during this developing the varieties with high protein content and quality. Generally, the mean protein content is 23% in case of red gram, and the wild species are having around 27 to 29%. We are having the two seed coat color in case of red gram. First one is a red seed coat color, and next one is white seed color. White seed coat. Red seed coat contains around more polyphenol, that is, more tannin will be present in the red seed coat than the white seed coat. So mostly we'll prefer the white seed coat varieties, and red grain contains lesser amount of the sulfur containing amino acids. When we increase the protein content, there will be decreasing amount of this amino acids also. So we have to so care is to be taken to increase this. Suppose if we increase the protein content, the amino acid content will be decreased. So we have to take care during this developing the high protein content. And next, coming to the breeding methods, the first breeding method is introduction. Through introduction, that is a Prabhat, a short duration variety, which was introduced from IARI. And next is ICPL 87 from ICRISAT. Next is pure line selection. Pure line selection is generally, before knowing that, generally earlier they thought it is a self-pollinated crop. During that time they have used the pure line selection, but later found it is a often cross-pollinated crop. So they have not used much more after that this pure line selection. And next is hybridization and selection. In here we are having the two types of hybridization, that is intervarietal hybridization, and next is intergeneric hybridization. Intervarietal hybridization, here we will be having, that is through intervarietal hybridization they have developed VBN1. It is a cross between Prabhat NY34 and T12 into 102. And next is intergeneric hybridization, that is a Khajanus khajan and Khajanus lineata, and Khajanus khajan and Khajanus caraboides are they are attempted through this intergeneric hybridization. And next is the fourth breeding method, that is the population improvement method. Generally we'll go for the recurrent selection method. For this recurrent selection method, we'll use two population. The one population is for the seed parent and other is for pollen parent. The seed parent must have one or two easily identifiable recessive characters, whereas the pollen parent should have dominant genes. So the seeds and the pollen parents are sown in alternate rows, so has to maximize the more cross pollination. Then the F1s and the selfed ones are identified in the S4 generation, and this identified F1s are space planted in the next generation, that is denoted as S1. And next in S2 generation, they are tested for the yield in 3 environments, and the best ones are recycled and then taken to the conventional breeding programs. And next, so these are the four breeding methods which are present in the red gram crop, that is introduction, pure line selection, hybridization, and selection. So the most important breeding centers which was working for this red gram is ICRISAT, that is International Crop Research Institute for Semi-Arid Tropics, which is located in Hyderabad. And next, coming to the practical achievements, varieties which was developed in the red gram are Prabhat, Vishaka, Sharada, ICPL 87. And next is hybrids are ICPH 8. So this is about the red gram crop. Here you can see, first we discuss about the introduction of the red gram, and here we have discussed about the varieties of the red gram. And next, these are the breeding objectives of the red gram, and next these are the breeding methods, and next breeding centers we have discussed, and practical achievements in red gram we have discussed. And next, coming to the green gram crop, this green gram is this is the most important pulse crop which having more digestibility when compared to the other pulses. So here the scientific name of the green gram is Vigna radiata, and is having the chromosome number 2n equal to 22. It is also diploid species, and mostly in the common words it is known as a moon bean. And the origin of the green gram is India, and distribution is it is grown in India, Pakistan, Bangladesh, Sri Lanka, Philippines, Taiwan, Thailand, Nepal, and southern Asian countries. And whereas in India, Maharashtra, UP, MP, Karnataka, Gujarat, Andhra Pradesh, Tamil Nadu, and Rajasthan, they are grown. Next, coming to the breeding objectives. The first breeding objective is the high yielding, short duration irrigated varieties. Generally, the lines having the rapid growth rate or dry matter increases associated with high harvest index. So generally, the green gram is having very around 3 to 4 months of the duration. So we can choose the high yielding varieties with short duration varieties for irrigated conditions. And next is breeding for disease resistance. Here the major disease in case of in case of green gram is yellow mosaic virus. Because of this virus, the entire crop will lead to the failure. And next is the leaf crinkle virus. And next is the for this, the Thai local LM214 is resistant to these diseases. And next is breeding for the quality. The quality is most important in case of pulses. So Mungbean, in the earlier I told that Mungbean is having the highest digestibility among the grain pulses, from around 83 to 90%. So the varieties having the bold seeds, we can use it as a sprouts. Sprouts is a major aim. And next is to transfer the high methionine content from the black gram to green gram. And next is high dal recovery, that is around 80% of the dal recovery we can observe, and more also we can observe, and the less hard seed is observed in case of green gram. And next coming to the breeding methods. The first breeding method is introduction. Through introduction, they have introduced the Pusa Baisakhi in case of green gram, and through pure line selection, they have developed the Co1, and through hybridization and selection. Here we are having the interspecific hybridization. This interspecific hybridization is done to transfer the high methionine content from the black gram to the green gram. And next is Vigna radiata is crossed with Vigna umbellata, being to transfer the resistance to bean fly, crossing with Vigna radiata variety Suplopata resistance to bruschetts. And next is mutation breeding. For mutation breeding, the Co4 mutant, the Co4 was developed. And next is embryo culture. Here they have crossed green gram into black gram through embryo culture. And next is ideal plant type. Generally, the ideal plant type, we have to go, mostly we'll select the varieties with a short duration, that is around 60 to 65 days with a determinate growth habit for irrigated conditions, and 80 days duration with indeterminate type for dryland conditions, and the plants with more pods and seeds, whereas increased branches podding from the base of the main stem with synchronized maturity and non-shattering habit, we have to select for ideal plant type. And the varieties they have developed in case of a green gram are, that is Jawahar 45, WGG2, and LGG 127. So this is about green gram crop, and that is here we have discussed about the introduction of the green gram. And next is the breeding objectives we have discussed, and next is breeding methods, and here you can see the varieties of the green gram. Next, coming to the black gram crop. The black gram crop, the scientific name of the black gram is Vigna mungo. The chromosome number is 2n=22, and for origin is India, and distribution is it is cultivated in India, Pakistan, Sri Lanka, and South Asian countries. Whereas in India, it is it is grown in Maharashtra, UP, MP, Karnataka, Gujarat, AP, Tamil Nadu, and Rajasthan. Next, coming to the breeding objectives, evolving medium duration high yielding varieties for dryland conditions. Generally the pulses mostly will grow in the rainfed areas. So we have to develop the varieties which are having the short durations with a high yielding for dryland conditions. And next is evolving the short duration high yielding varieties which are suitable for the dryland conditions. Generally we can use as a mixed cropping with cotton and turmeric. For this, the short duration varieties are Co2, Vamban 1, 2, and 3. And next is breeding varieties which are resistant to the diseases. Here also, in case of green gram and black gram, yellow mosaic virus is a serious disease, whereas leaf crinkle virus and powdery mildew also we can observe in case of black gram. And next is breeding for pest resistance. The major pest in case of a black gram is it is a white fly, which acts as a vector for the yellow mosaic virus, and leaf crinkle and leaf eating caterpillar. So these are the major pests which are prevalent in the black gram. And next is breeding for better quality. Generally, the black gram is having around 24% of the protein content. So those lines which are having around 27% of the protein, which can be utilized. So here the quality of the black gram is determined based on the protein content, methionine content, that is 1.17%, and next is based on the cooking quality, based on the time, based on the duration of the time we can check the we can we can decide the quality of the black gram. And next is the percent of the hard seeds. And next is dal recovery, around 70%. Out of all pulses, generally the green gram is having more dal recovery, that is 80% of the dal recovery we can observe. And next is coming to the breeding methods. The first breeding method is hybridization and selection. Here we are having the two types of hybridization, that is intervarietal hybridization and interspecific hybridization. Interspecific hybridization, here you can see Vigna Mungo and Vigna Mungo variety Sylvestris, which was crossed at Pantnagar. This yellow mosaic resistant lines are through which the yellow mosaic resistance are mosaic resistant lines are obtained. But they are the pod shatterers, so more number of back crosses are suggested. And next is Vigna Mungo is crossed with Vigna radiata for increasing the pod length as well as digestibility. And the sterility is a major problem, and a few plants obtained which are revert back to the parental forms. And next is mutation breeding, and next is embryo rescue. They have attempted in some of the interspecific processes. And next is breeding centers. Here we are having the three centers which are working for black gram. First is Sikrisat, that is located in the Hyderabad. Next is International Crop for Agriculture Research in dryland areas, which is located in the Syria. And next is Indian Institute of Pulse Research, that is Kanpur, which is also working on this black gram. These are the three breeding centers which are working on the black gram. And next here you can see the black gram varieties, that is T9, T27, LBG17, LBG 402. So these are the varieties of the black gram. Next, coming to the so this is about the black gram crop. Here we have discussed about the introduction of the black gram. The scientific name is Vigna Mungo, and here we are having the chromosome number 22, and the origin is India. And here you can see the breeding objectives. And here the quality is more important in case of black gram. So this was the quality black gram is determined by based on the protein content, methionine content, that is 1.17%, and based on the cooking quality, and based on the percent of hard seeds, and based on the dal recovery. And here you can see the breeding objectives. So this is about the black gram crop. And next, coming to the soybean. Um, Soybean, as we know, the soybean is a miracle crop. The scientific name of this soybean is Glycine max. It is having the chromosome number 2n equal to 40. It is also a diploid species, and the origin of the soybean is China. And next is distribution. Distribution is it was grown in USA, Brazil, China, Argentina, and India. And the progenitors for this are Glycine. Userensis and Glycine tomentola, Glycine tabacina, and Glycine gracialis. Generally, the Glycine max is this is originated from Glycine userensis and Glycine tomentola. The cross between these two, we have obtained the Glycine max. And generally, this soybean also is the most important. This soybean is grown as a pulse crop, that is a legume crop, as well as it is the most important oil yielding crop of the world. So it is called as a miracle crop. It gives around 42 to 45% of the protein content, and around 19 to 20% of the oil, and it belongs to the leguminaceae family. So this is about the introduction of the soybean. That is the scientific name is Glycine max. It is having the chromosome number 40, and origin is China, and it is obtained from cross between Glycine userensis and Glycine tomentola, and it is the most important oil yielding crop of the world. So it is called as a miracle crop, and it is having around it gives around 42 to 45% of the protein content, and 19 to 20% of the oil content. It belongs to the leguminaceae family. So next, coming to the breeding objectives. The first breeding objective is breeding for short duration high yielding varieties. Generally, the yield in the soybean plant is determined by the size, next number of the seeds per pod, and number of pods per plant. Based on these three parameters, the yield of the soybean plant is determined. And out of that, the number of the pods per plant is determined by the number of nodes per plant and number of pods per node. So each of the above components of the yield are polygenic in inheritance. So it is complex. So the development for yield is governed by the polygenic character. So the yield improvement is very difficult in case of soybean crop. So the duration is also determined by the multiple genes, and maturity is correlated with the height of the plant, and the early varieties will be the short in stature. So this is about the breeding for short duration high yielding varieties. Here the yield is determined by the yield of the soybean plant is determined by the size, number of seeds per pod, and number of pods per plant. And number of pods per plant is determined by number of nodes per plant and number of pods per node. So here the yield in soybean is governed by the polygen. This is a complex character. It is difficult to improve the yield in case of soybean. And next is breeding varieties which are suitable for the rice fallow crop. Generally, the short duration varieties around the 65 to 70 days duration we have to select, and which are suitable for the intercropping also, and suitable for intercropping in case of banana and sugarcane crop. And next is breeding for the quality. Generally, the seed coat color and the quality. Here we are having the seed coat that is yellow color, black color, and brown green color. And next is the oil content and quality and protein content. So these are the three important quality parameters which can observe in the soybean crop, that is seed coat color, oil content, and the protein content. So first is the seed coat color. The seed coat color may be, generally the seed coat in soybean may be yellow or green, black or brown, or combination of all these colors. So for oil extraction, the yellow color is mostly preferred because a high oil content is present in the yellow color, whereas the black varieties are having low in oil content, but they are having high protein content. So this is about the seed coat color. It may be the seed coat color may be yellow, green, black, brown, or a combination of all this color. But for oil extraction, we'll use the yellow seed coat color, which is having the high oil content, whereas black seeded varieties are low in oil content but high in protein content. And next is oil content and quality. Oil content is generally determined by the environment. Yellow seed coat colors are rich in oil content, and the complex character is determined by the polygenes. And next is protein content and quality. Earlier we have discussed that soybean is having the more protein content, that is around 35 to 50% it is having. The protein content is negatively correlated with oil content. So if we'll improve the protein content, the oil content will be reduced. If we'll improve the oil content, the protein content will be reduced. So this protein content and oil content are negatively correlated. So breeding for high protein content is a compromises to be made. And next is breeding for vegetable type. That is this is a center, that is Asian Vegetable Research Development Center, which was located in the Taiwan. They have developed some of the vegetable types in case of soybean. And next is breeding for non-shattering type. Here you can see the varieties Lee and Co2. And next is breeding for yellow mosaic resistant lines. Here we are having the Co2 line. And next, coming to the breeding methods, here we are having the three methods, that is introduction, pure line selection, hybridization, and selection. So the first one is introduction. EC39821, it is from Taiwan, which is released as a Co1, and through pure line selection they have developed the Co1, and through hybridization and selection they have developed the Clark. And next, coming to the breeding centers which are working on the soybean, that is AVRDC, that is Asian Vegetable Research and Development Center, which is located in the Taiwan. And next is International Institute of the Tropical Agriculture, which is located in the Nigeria. And next is Indian Institute of that is in India, the National Research Center for the Soybean, which is located in the Indore, MP. So these are the three institutes which are working on the soybean crop. And next is practical achievements. Here you can see the varieties of the soybean, that is Bragg, Clad 63, Hardy, and Max 13. So the summary is, generally the pulses are more protein rich crops, but generally they are low yielders than the cereals. They provide the good amount of the lysine, tryptophan, and threonine, but they are low in sulfur containing amino acid, that is methionine, cysteine, and cysteine. but they are more in the case of cereals. And the major pulses it includes are PGNP, that is Kajanus Kajan, green gram, that is Vigna radiata, black gram, that is Vigna mongo, and soybean, that is glycine max. And the breeding object is mostly focused on the high yield, short duration, bold grain vegetable types, pest and disease resistance, that is yellow mosaic virus, sterility mosaic, and root rot, and improved protein and oil quality in case of these pulses. And next is methods. Methods we have mostly concentrated on the pure line selection, hybridization, mutation, recurrent selection, and interspecific hybridization. And the key institutes which are working on this pulses are ICRISAT, IIPR, that is Indian Institute of Pulse Research, and NRCS, that is located in located in Indore. And next is AVRDC, which is located in the Taiwan, and IITA, which is located in the Nigeria.